Hello, my name is Mara Costello. I'm a molecular biologist at the Technology Development Group here at the Broad, and I'm going to be talking to you about sample preparation today. So in this presentation, I'm mostly going to be talking about genomic DNA processes, because that's mainly what we do here in our group at the Broad. But a lot of these techniques can be applied to different sample types. However, mainly I'm going to be talking about genomic DNA. Sort of the Broad's best practices and some tips and tricks that we like to employ. First, we're going to go over Broad's sample preparation workflow in general, how we track samples through the lab, and some Broad-developed modifications to sample preparation and how we've applied these to our protocols. These include different shearing methods, enzymatic reaction cleanup methods, size selection methods, both with gels and without, and PCR enrichment protocols. And then finally, go over some quality control methodology for sample prep, including an overview of some sequence metrics that you can use to assess sample preparation quality. So this here is our general sample prep workflow at the Broad Institute. It's really not that different than what the standard Illumina workflow is. So all the areas in red are the places that we at the Broad have made some modifications, and we found some various ways of improving our process and making it more streamlined. First thing we're going to talk about is sample tracking. So here at the Broad, we track our samples all the way from the very beginning, from where we actually get our genomic DNA from our sample repository group. So in our high throughput world here at the Broad, where we're processing hundreds of samples every day, we really need to have an ironclad method of tracking all our samples through the lab. The most important thing that we've implemented are the use of these 2D barcoded tubes. We store all our DNA, including our source genomic DNA, and all intermediate library types, including unenriched, enriched, and normalized. All of these have 2D barcodes on the bottom of the tube and allow us to track their movements through the lab and to associate different tubes with each other. All of these transfers and tracking information is detailed in our web-based LIMS system. And you can see here in the lower right, there's an example of our LIMS system here. And you can see that we can track our samples all the way from the genomic DNA number down to the actual lane of what flow cell it's been on. We also have implemented a tube picking system, which is in the instrument on the lower left here. It allows us to select the proper libraries based on their barcodes to help error-proof flow cell loading. But at lower throughputs, it's also important to track your samples. Before we sort of scaled up to the level we were at, we used to just track things with Excel spreadsheets, which we also found extremely helpful. Implementing a library numbering system allows for easy tracking, even if you don't have a barcode reading system. Also, keeping an Excel tracking sheet can help you associate different libraries with each other and also help you with downstream troubleshooting. For instance, if you can track that one particular technician maybe was having issues with making libraries, you'll be able to sort things and find it out that way. Also, keeping track of where libraries are stored in a freezer system is very beneficial for if you have to go back and rerun a library, you'll be able to find where it is easily. Next, we're going to talk about how we shear our genomic DNA here at the Broad. So at the Broad, we do all of our shearing for our standard Illumina library construction using an instrument called the Covaris. So Covaris is a company that was founded in 1999, and they developed a line of instruments that provide controlled delivery of acoustic energy to closed vessels. And originally, they didn't really target the biotech industry at all. They targeted the chemical industry for pulverization and homogenizing of chemical compounds. But we were the first here at the Broad to work with Covaris to develop their method and apply their adaptive-focused acoustics technology to the process of DNA shearing. And our main reason for moving to the Covaris was the throughput that it provided. With the E210 model, you can shear up to 96 samples at once. So the way Covaris works is you've got on the right-hand side here, you can see a diagram of what's going on actually in the Covaris. So you've got a water bath, and you've got a transducer here, which is a metal bowl that shoots the acoustic energy at the sample in the glass vessel. And this acoustic energy causes cavitation, or basically a rapid vortexing action within the tube. And it's this rapid creation and popping of these bubbles that leads to the mechanical shearing of the DNA. And as I said, the E210 model, which is the one on the lower left here, is for 96 samples at a time. You can do up to 96 samples, anywhere from 1 to 96. They also have a single tube-based model, which is the S2, which is the one to the right, and that is for lower throughputs. So we've worked out a lot of conditions here for the Covaris for shearing DNA at different sizes. I've listed some here in the table. We've got a validated 150, 200 base pair, and also a 300 to 700 base pair shear for larger inserts. And you can see some examples of these shears at the bottom. So you'll see more examples of these data and how you can manipulate these parameters to get different size shears in the laboratory section of this program. So choosing a DNA shearing method definitely depends on sort of what your throughput is and what you sort of want to spend for your laboratory equipment. So the nebulizer that comes with the Illumina sample prep kit can give you DNA in the desired range for Illumina library construction, is reproducible, is customizable in that you can sort of limit, it's a little bit limited, but you can vary the time and pressure to get DNA sheared to different sizes. The main benefit is that there are no additional materials costs because it comes with the kit, and you also don't need any additional instrumentation. All you need is some house air, basically. The Covaris instruments also give you DNA in the desired range or reproducible. They're more customizable because you have 5 different settings that you can vary. But the drawback with them is that they do require significant capital investment because they're, instrumentation. But for the E210 model, the reason we chose it is because it can shear multiple samples at a time. And because it's in a 96-plate format, it's compatible with our automation that we have here. So basically, choosing a shearing method comes down to what your particular lab is looking to achieve. Next, we're going to move on to reaction cleanup between all the enzymatic steps. So the reaction cleanups, there's basically 2 main types of sample cleanups. There's either the column-based, such as the QIAGEN Minilute columns, or a magnetic bead-based cleanup method. The column-based are reliable and reproducible. They give you about 80% recovery. They're very easy to perform. But one of the drawbacks is that they're really only ideal for lower throughputs. They're not really compatible with automation. And so at least here at the Broad, we say that a reasonable set size is less than 12 samples because you really don't want to be handling that many individual tubes because it sort of increases the chance of sample swapping. Magnetic bead-based cleanups are also reliable and reproducible. You get about 80 to 90% recovery as well, and they too are easy to perform. But one of the benefits of them is that they can also be automated easily, or they can be done manually. So manually, you can do 8 to 12 reasonably in a tube by hand. But automated, if you had a 96-well magnet and a liquid handler, you can do plates of these at a time. And because of this reason, the Broad has moved away from column cleanups to the magnetic-based cleanups as much as possible, just to sort of improve our throughput. So the magnetic bead-based cleanup that we chose here at the Broad is called SPRI. It's a product by Agincourt. Its brand name is Ampere. And SPRI stands for Solid Phase Reversible Immobilization. And it's a technology that, again, it's magnetic bead-based, and it's specific for double-stranded DNA. It only binds the DNA, and any excess primers and enzymes or anything else that you don't want is washed away, and then your DNA is eluted. And I've included the reference here for the original paper for the SPRI technology if anyone is interested in reading up more about the details of how it actually works. But in general, the way it works, and we'll go through this diagram here at the bottom. So you have your DNA sample. The little blue helices here, those are your actual double-stranded DNA that you want to purify. And the brown triangles are anything you don't want. It's that enzyme in the primer. You then add your SPRI beads, which are the little red circles. That binds the double-stranded DNA. You then separate that on a magnet. So the magnetic beads pull the DNA down, leaving all the unwanted enzyme and primer in solution. You then remove the supernatant, wash it with ethanol, and elute with elution buffer. And you end up with cleaned up DNA at the end. And as you can see, because it's just liquid transfers, it's very amenable to automation. So another property of SPRI that can actually be manipulated is that you can vary the size of fragments that bind to SPRI by varying the amount of SPRI that you add to the reaction. There's something called the X of SPRI, that refers to the volume-to-volume ratio of SPRI to DNA. So, for example, a 1X SPRI is a 1-to-1 volume ratio of SPRI to DNA. So if you have a 50 microliter reaction, you add 50 microliters of SPRI, that is a 1X SPRI. What you can see in the gel image here at the right is that the lower the X of SPRI, so the lower your ratio of SPRI to DNA, the higher the size cutoff becomes. You start to lose the smaller and smaller fragments, the less and less SPRI you add. And we find that a 0.7X SPRI removes fragments below 150 fairly efficiently. And that's basically where a lot of your adapter dimer lives. But you can see that there is a cutoff, that between a 0.5x SPRI and a 0.4x SPRI, you almost lose everything. So there is a cliff there that you can't really go past. But we find that anything between a 0.5 and a 2.5x SPRI can be manipulated to sort of do a crude sizing. And as we'll see in a few slides in the size selection section, you can use this property to do a sizing, and you can actually do what we call a double-sided SPRI, which I'll talk about later, to sort of get rid of both high and low fragments this way. So how we've applied SPRI is to our high throughput libraries. Because it can be done in a plate, it allows us to do libraries at a much larger scale than we were ever able to before. And one of the challenges we had is that for a lot of projects that didn't need tight sizing, that just needed, some coverage-based projects, they wanted 96 libraries a week or more. They had 3 micrograms of DNA to start with. And again, they didn't need gels. They just wanted libraries that they could get coverage just run on the sequencer. And so by implementing our Covaris shearing optimized to an insert of about 200 base pair and the automated SPRI cleanups on the Velocity 11 Bravo liquid handler, we can make libraries that have an insert of 250 base pair and have no adapter dimer because it was removed with the 0.7x SPRI. An example here of the final Agilent after PCR is shown in the Agilent picture on the lower right. These are pretty consistent yields, pretty consistent sizing as well. And these were done basically in a day because there's no gels needed, no column cleanups needed. You just shear and go straight through enzymatic reactions and SPRI on the automation. So next we're going to talk about size selection methods. So at the Broad, we kind of have 2 different paths that a library can go down. It's either what we call a gel-free library, where it goes directly from the enzymatic steps and adapter ligation through a SPRI cleanup and then into PCR enrichment, or we have gel libraries where they have to go through an Agarose Gel size selection before PCR enrichment. So before I talk about the various methods for size selection, I wanted to sort of have an aside here to just talk about the difference between insert size and library fragment size. These 2 terms kind of get thrown around a lot, and I know we do it as well here at the Broad, and it's good to just get sort of a definition of what people are talking about when they say these 2 different terms. Your insert size is actually just the length of your genomic DNA insert. So in this example here, we've sheared and size selected, we've sheared a DNA piece to about 300 base pair. So that's your insert size, 300 base pair. Then after paired end adapter ligation, you're going to add 33 base pair per side because that's how long your adapters are per side. So now you've got a total of 366 base pair. So if you're going to do gel extraction after your adapter ligation, you actually want to cut not 300 base pair, but you want to cut at 366 base pair in order to take into account that you've just added your adapters onto the end of your fragment. Now you're going to do an enrichment, and that actually adds more bases on. So your paired end PCR enrichment is going to add 28 base pair to that P5 end and 25 base pair to the P7 end. So now your completed fragment is 419 base pair. So that's what, when we say library fragment size, that's what that final piece of DNA that you're going to put on the flow cell is. It's 419 base pair. But really, the insert is only 300 base pair. So we've definitely made that mistake here at the Broad where somebody asked for 300 base pair insert and we make a 300 base pair fragment. But now your insert's actually 119 bases shorter than that, so it's really not what the collaborator wanted. So I just wanted to sort of point that out because I know we get confused with that a lot and wanted to sort of throw that slide out there. So choosing a library fragment size is also an important thing to keep in mind. A library fragment size of about 400 base pair is what Illumina says is optimal for cluster density and for the sequencing optics and cluster finding algorithms. But the sequencer can actually tolerate fragment sizes all the way up to about 800 base pair. So really your fragment size can be tailored to suit the needs of any project. But something to keep in mind is that your insert size is dependent on your read length. You don't want it, for long read lengths, you don't want an insert size that's too small because you're either going to read into your adapter on the other side or you're just going to read the same piece of DNA in both directions. So for 36 paired sequencing, your insert should be at least 72 bases, if not greater. And for 76 paired, it should be at least 152. Just something to keep in mind to sort of make sure that your paired sequencing is actually getting you what you want. And another caveat to keep in mind is that larger fragments are going to make larger clusters in the flow cell. And that's going to sort of limit what your maximum density is because you can't pack them in as tightly because they're sort of larger, fatter clusters. And another thing is that a wider range of size distribution is going to also make a wider range of cluster sizes. So you're going to have very large ones next to very small ones, and that too is going to lower your maximum density that you can achieve because, again, you can't pack them in because they're not uniform. So once you figured out what your insert size is going to be, next you have to figure out how you want to get there. So here at the Broad, we basically have three options for size selection that we choose from. Option one is your regular old Agarose Gels, which I'll talk about sort of some of the conditions we have for that. Option two is something we call double-sided SPRI, which I'll go through, sort of utilizing those properties of SPRI that allow you to sort of bind preferential sizes. And then option three is no size selection at all, which if you have a tight shearing, you don't really need it for a lot of projects. And I'll go through all three of these. So option one is Agarose Gels. Oftentimes, size selection is necessary. You can't really get around it if you want to have size deviations that are less than 30%. And the Broad's recommendations for gel conditions are shown here. What we found is that for small fragments that are less than 400 base pair, we find a 4% new sieve 3-to-1 gel run at 85 volts for 3 hours at room temp. Does a really good job at resolving those smaller sizes. And then for stuff that's greater than 400 base pair, up to about 1 kb, we actually run a 2% low melt gel with something called C-plaque agarose. And we run that a little faster at 110 volts, also for 3 hours, but we do this in a cold room, just to make sure that that low melt gel doesn't lose its integrity. And that does a fantastic job of resolving things right around 500 to 600 base pair. We can also get very tight size distributions of 10% or less by taking very, very thin gel slices. In the image here at the bottom, you can see this is an example of we were targeting a 409 base pair fragment. And these gel slices are about 1 millimeter thick, and we were able to get plus or minus, I think that this was 5% here. And in the lab section, you'll see a demo of a gel cutting tool that we've developed that allows you to take these very, very uniform thin slices, and it makes taking these tight cuts a lot easier. So how do we apply these tight cuts to our library construction efforts? One of the library types that we make is something that we call a 180 base pair library. This is actually requested by our assembly team, our informaticists, who wanted us to make libraries that had inserts that were nearly exactly 180 base pair. The reason they wanted to do this, you can see a diagram of it here in the upper right, is that if you have a 180 base pair insert and you do a paired 100 base pair run, you get a little tiny overlap in the middle. And what that does allows them to align those paired reads as one giant 180 base pair virtual read, they call it. And longer reads are more robust for assembly. So it basically allows them to reap the benefits of an almost 200 base pair read when really on Illumina we do 100 base pair reads. To accomplish this, we take 3 extremely tight gel slices at about 246 base pair in the gel. So that's after adapter ligation, which is 180 plus 66. That's where we get the 246 base pair from. You can see an example of this in the gel image here at the bottom right. So we use a gel cutting tool that holds 4 razor blades about 1 millimeter apart. We then take these 3 gel slices, prep them all, and after PCR, run an Agilent chip. And we select the one that's closest to 299 base pair, which that is because your final fragment is 119 plus your insert. That's pretty much exactly 180 base pair. And our assembly team has been very happy with these results. They use these to do de novo assemblies of a lot of bacteria as well as mammals. So they've been really useful for us. So option 2, if you don't need sizing that's super tight, but you need some kind of specificity there, is something that we call a double-sided SPRI. So as I previously showed, and I've got this gel picture up here again, you can sort of vary the size of fragment that SPRI binds by varying that X of SPRI, so that ratio of SPRI to your DNA. And we can utilize this property to perform a gel-free and automated size selection. So I'm going to go through this now. It's a little bit confusing, so I'm going to go through it kind of slowly. What we've done is we've implemented a combination of good shearing right at about 150 base pair. And then we do sort of 2 serial SPRIs, which I'll go through how we do those. And we can select for a fairly tight size range without a gel. So the first SPRI we do is a 0.65x SPRI. So we've got our DNA, we've sheared it to 200 base pair, and we're going to do a 0.65x SPRI. What that does is binds fragments above 250 base pair onto the beads. and leaves everything below 250 in the supernatant. But instead of keeping the beads like we normally would do, we're going to keep the supernatant and throw away the beads. So what that does is now we've kept everything 0 to 250. And because we've chucked the beads, we've gotten rid of the things that are above 250 base pair. We then take that supernatant and add some more SPRI. This is a larger X. It's a 1X SPRI. And what this is going to do is bind everything above 100 base pair on the beads and leaves everything below 100 base pair in the supernatant. And now this time we are going to keep the beads. So we're going to throw away that supernatant that has all the small stuff in it and keep the beads, which now has everything 100 to 250 base pair bound. Here's an example of our results. The red curve here is the DNA profile after shearing. So we got our 200 shear. It's not bad, but it's, still got some stuff that's, out into 500 base pair and down to about 50 base pair. So the green curve here is the result of this 0.65x 1x double-sided SPRI. You can see that the first SPRI, the 0.65x, got rid of everything that was between 250 and 500, and the 1x SPRI got rid of all the lower molecular weight things that were between 50 and 100 base pair. But what's good to notice is that you didn't take a large hit in yield. The curves are still fairly robust here, even though we've done 2 different SPRI cleanups. And then this blue curve here just shows that if you play with these X's, you can actually kind of change what the size selection, what your final product is. By lowering the X of SPRI a little bit more on that first X, 2.55X, we were actually able to make our sizing a little bit wider. So we included fragments now out to about 300 base pair. Again, as I had said before, there is a cutoff there with SPRI. If you go any farther below about 0.5x, you end up losing almost all your fragments. So there's not, a huge range of sizing that this works on, but you can definitely play with this and get sizing pretty much anywhere between 100 up to like 300 base pair if you just sort of play with these ratios. So option 3 is if you just don't do any size selection at all. If you have a tight shear from Covaris or even nebulizer, if you've got your nebulization conditions optimized, you can get away with not doing any size selection at all. For most libraries that just want coverage, a lot of projects, they don't need super tight sizing. They're not looking for structural rearrangements or indels or anything like that. They just want some coverage. So those type of projects we put through our size selection-free pipeline. And what we do is we combine 150 base pair shear and a 0.7x SPRI. So you end up with fragments that have inserts of about 150 to 200 base pair, and that low X SPRI, the 0.7x SPRI, has gotten rid of all of your adapter dimers, so that's not a concern. So really, you don't need to do any gel at all. So some examples here are shown in these 2 Agilent profiles. So the first profile is 150 base pair shear. This is the Agilent post-shearing. And then we took the entire volume here through N-repair, A-addition, adapter ligation. We didn't do a gel, but we did a 0.7x SPRI. And then we took 1 microliter of that 30 microliter SPRI elution and did 10 cycles of PCR. And these here are the resulting Agilent curves from that. So you can see, even though we only took 1/30th of the sample, we still have a fairly robust peak here. And it's very consistent from library to library. And the sizing is about 200 to 400 base pair, which for most of our projects seems to be perfectly acceptable. So this kind of process has allowed us to just churn through libraries, make hundreds of these basically in a day, because this whole process can be done in about 8 hours because there's no gels. As you can see, these are fairly robust because, again, we only took 1/30th of that DNA and put it through enrichment PCR, and we still have a pretty good yield. We could do 30 or 29 more of these PCRs and get 29 times the yield here. So plenty of DNA, great for high coverage projects where you have to run lanes and lanes and lanes. So now that you've size selected your sample, the next step is to PCR enrich the sample. So one important point to think about when it comes to PCR enrichment is this idea of library complexity. And that refers to the number of unique fragments that are present at the end of library construction in your given library. And complexity can be affected by a whole bunch of things: how much DNA you started with, how much you lost in all your cleanups. If you have to do a gel, you're going to lose a lot of your DNA just through gel size selection and extraction. And then the amount of duplication that you've introduced in your PCR. Because basically PCR is just making copies of things. And if you start with not that many copies of something and you do many cycles of PCR, you're going to end up with a fairly highly duplicated library. For most libraries that only need to be run across a few lanes, the standard Illumina protocol provides libraries with ample complexity, and that's, I think, usually 1 microliter at 18 cycles of PCR. But for certain projects, especially for very deep coverage libraries where you have to get to 30x, 60x with one library complexity becomes more important. And a lot of projects such as SNP discovery, mammalian assembly, and cancer resequencing kind of fall in this category. So there is a big relationship between PCR and complexity. And I'm going to walk through, this is an experiment that we did looking at how PCR can affect the duplication that you see in the final library. What we did here is we varied our input volume of DNA to the PCR, the number of PCR cycles that we did, and the number of reactions that we pulled together. And we set up these PCRs and then sequenced them and determined the percent duplication, and based on that, our informaticists can estimate the total number of unique fragments that are in the library itself. What we did was, based on this data, we were able to estimate, basically, for each new lane that you sequenced, how many new molecules would you keep seeing? So, in other words, if in a perfectly 100% diverse library where there were infinite numbers of fragments, you would keep seeing new fragments every time you sequenced a new lane. But in reality, we know that that's not going to happen, so you're going to end up plateauing eventually. But how soon you hit that plateau is sort of indicative of how duplicated your library is. And that data was graphed in this graph you can see here at the right. What we've got here is we found that our original PCR conditions, which are the red line here in the graph, where we did 1 microliter of DNA into our PCR, we did 14 PCR reactions in just one reaction. You can see this line started to plateau fairly early. And again, as I said, for 1, 2, 3, even 4 lanes of sequencing, it really doesn't make a huge difference. But when you start going out to 8 lanes, 10 lanes, you can see that we're plateauing out here. And eventually, we're never going to sequence any new molecules after about 10 or 12 lanes, I believe, on this type of library, which is, not ideal because for a lot of our 30 and 60x coverage, we have to run dozens of lanes. So then we said, what if we just reduce our PCR cycle number? Maybe we're just making too many copies of everything. So that's what this blue line is here. You can see that that did help. Just reducing the cycles from 14 to 10 did reduce the duplication. But again, you're still plateauing out because there's only so many unique fragments that are in there. What improves the most here, and this is the yellow and the green lines, is that when you put more material into your PCR, which makes sense. The more you start with, the more you're going to end up with at the end. The green line is when you put 6 microliters of DNA into 1 PCR at 10 cycles. And now you can see that's almost up at y equals x, which is nice. But the best result we got was when you actually pooled multiple reactions together. So that's the gold line here. And we did 6 reactions, each with 1 microliter of DNA and 10 cycles of PCR. You can see that that line isn't plateauing at all. And if you look down at the table below, you can see that the duplication percentage between the red and the gold here went down almost tenfold. We were at 5.5% duplicated for our original PCR conditions. And just pooling 6 cycles and taking down the cycle number by 4, we were able to get our percent duplication to below 0.5%, which means that this library can be sequenced on dozens and dozens of lanes and really not plateau out. So how we've applied sort of these findings is to what we've termed our high-diversity sample preparation method. Mainly this applies to the cancer resequencing projects that we've gotten. Many of them need 30x unique coverage from tumors and their matched normals, and we were having trouble delivering that 30x coverage before. What we needed to do is find a way to reach that 30x unique coverage with as few lanes of sequencing as possible, but also with as little genomic DNA as possible. We couldn't make 10 libraries from every tumor because we didn't have that much DNA. So what we needed to do was in that one library get the highest complexity with the lowest percent duplication. But because they were using a lot of this data to do indel detection and structural rearrangement detection, they requested that we size these libraries 500 base pair plus or minus 10%. So take a tiny, tiny piece of DNA, but try to get billions of molecules out at the end. So to accomplish this, we developed this high-diversity protocol. So what we do is we shear our DNA to about 400 base pair, and then on the gel, take 2 extremely tight gel cuts from each sample. We take one that's 500 base pair to 520 base pair, and then one that's right above it that's 520 to about 540 base pair. And what this does is it creates 2 libraries from every 3 microgram genomic DNA input. So we're basically doubling the amount of information we can get from one input. So we then gel extract those 2 slices separately and enrich the entire sample, the entire amount of both those cuts to maximize the diversity. And how we do that is we do 16 50 microliter PCR reactions with 10 cycles of enrichment. And then we pool all those reactions together and clean them up. So it's basically for every 1 3 microgram sample, we're doing 32 different PCRs, 16 PCRs for one cut and 16 PCRs for another cut. So here are some of the coverage results for these type of high diversity libraries. You can see the red circle here. We've got, on this graph here, the x-axis is PF aligned coverage. The y-axis is PF aligned unique coverage. So they took out the duplicates and now they're only looking at the unique coverage. And even with doing 16 PCRs per reaction, we still weren't hitting 30x unique coverage with 30x aligned. You can see it's a little shy there. 30x aligned really only equals 28x unique. So still not good enough. We would have had to sequence 2 more x, which is, money wasted basically. So that's kind of why we moved to taking both gel cuts and running both gel cuts. So the purple circle down here, you can see at 15x aligned coverage, you're at pretty much 15x unique. So you're right on the money here. And so what we've done is we take those 2 gel cuts and we run both gel cuts to 15x instead of trying to run one to 30x. We then aggregate that data together, and now the informaticists have their 30x unique coverage, but we really only had to run 15x plus 15x of aligned coverage. So it saves us sequencing money. It makes library construction harder, but it saves on what the most expensive part is, which is the sequencing. So finally, I'm going to talk about quality control for library construction. We do quality control at various points throughout the process, but in particular after DNA shearing and also at the end after PCR enrichment. Our main sort of workhorse for QC is the Agilent bioanalyzer, which I've shown some examples of this throughout the presentation. But we use the Agilent to ensure that our shearing works correctly, so we run it right after shearing. And then we also run it after PCR at the end of library construction to make sure that our sample is the correct size. And any failing samples can enter a rework loop, so we catch it right away. If it didn't shear properly, we can either re-shear it, or if it's completely the wrong size, we can ask for another aliquot. Or the same with the enrichment. If we have leftover material, if it either didn't enrich or we over-enriched or under-enriched, we can go back and start that over. So we kind of can catch problems before they hit the sequencer that way. Another QC we run is we do a pico-green quantitation post-enrichment. And similar to the Agilent, can kind of tell us if we have enough material. We actually like to run both because we like to see that the size is correct. And then we run the pico because we want to make sure we have an accurate quantitation of what we have there. The guideline we've set up for ourselves is that samples that are below 20 nanomole, we re-enrich if there's more material to go back to, or say in the case of the high diversity libraries, if that library hasn't hit 20 nanomole and you did 16 PCRs, there's probably something really wrong with it. So that would have to get restarted from the very beginning. We also take before and after gel pictures for any libraries that require gel cuts, and we save all these images. The reason we do this is it's good to, A, make sure that the technician has cut the proper size if a question comes up at the end of library construction that the library isn't the correct size. But also we can troubleshoot any weirdness, such as extra adapter dimer or DNA in unexpected size range. We've got an example here of how we were able to use the gel pictures to kind of post-mortem diagnose what went wrong with this library. So these were high-diversity libraries that we somehow reached the sequencer. We kind of missed these in our QC. And they had really high levels of duplication. And we weren't sure why because we had followed the protocol to the letter. When we went back and looked at the gel images, we can see that up the top of the gel, there's a lot of unsheared material. So the shearing wasn't 100% efficient for these samples for some reason, and so most of your material was not in the size range that you cut. So that sort of explained why there was low duplication. But if we hadn't saved these gel images, we wouldn't have been able to figure that out. Finally, you can actually use sequencing metrics to look at the quality of your library after it's been run. And of course, I just want to say that you should always check that your actual sequencing quality metrics were okay before looking at these LC metrics. There's no point in trying to figure out if your library was good if your sequencing run actually was bad, if your error rate was high or you had weird phasing or pre-phasing or any of those sequencing metrics are off. But if you do have a good sequencing run, you can look at these various metrics to figure out how good your library is. The most basic one is just the percent aligned, and that's the percent passing filter reads that align to your reference sequence. And a low percent aligned may indicate a sample contamination or a swap. In other words, what you think is in that lane may not actually be it if it's not aligning to your reference sequence. So the following 4 metrics here are metrics that our Broad pipeline spits out for us, but I believe Illumina does have scripts to determine all of these different metrics that you can get from them. The first one is the percent adapter. That's the percent of passing filter reads that basically align to the Illumina adapter. And our cutoff we have is that we say if there's greater than 1% adapter, it indicates that something went wrong with LC most likely, or library construction, most likely. You either had inefficient removal of adapter, or you had too much adapter, something happened. Another metric we look at is percent chimerism. And that basically is the percent of reads that have 2 ends that are over 100 kb apart or on 2 separate chromosomes. So basically it's a paired read that the 2 reads have no business being on the same molecule, basically, yet somehow they are. And percent chimerism can be indicative of a couple of things. Could be a problem in adapter ligation if you ended up, instead of just putting one insert between adapters, if you have 2 or 3 inserts that somehow get sandwiched between one set of adapters. Or it could be a problem with genomic DNA prep itself. If these samples were whole genome amplified or had some other processing, there could have been some weird structural rearrangements that were artificially introduced. Percent duplication and estimated library size I talked about earlier when we were talking about PCR, but these are an indication of how duplicated your library is. So for percent duplication, it's your read 1 and read 2 for 2 different clusters have the exact same start sites and end sites. So basically, it is the same molecule. An estimated library size is something that it's a kind of complicated calculation, but it's based off the percent duplication as well as how big your genome is. For example, human is much larger than E. coli. You'd expect much more just naturally duplication if you were running a small bacteria just because it's a much smaller genome, whereas with human, you really shouldn't, if you've got good representation, see that much duplication. And what we found is for our whole genome shotgun libraries where we do the high diversity preparation, we should have a library size of 1 to 3 billion molecules. If it's any lower than that, it might not be able to reach that 30x or 15x coverage. 2 other important metrics to look at are concordance and insert size plots. So the concordance is the concordance of sequencing data that you've received with previous genotyping or SNP data. And this can be useful to determine sample swaps. So if you have a SNP panel run on a sample, and when you look at your sequencing, it doesn't make sense. With the SNP panel, it might have been swapped somewhere in library construction. So insert size plots are also useful, and it shows just the size distribution of your sequence fragments. It can show if you have a size selection problem, like for instance, the image here, we have a low molecular weight shoulder, so it looks like our gel conditions need to be tweaked. But it can also indicate sample swaps as well. If you expect that your library is 400 base pair and it's showing on your sequencing that it's only 200 base pair, it might not be the library you think it is as well. In summary, I hope that you got from this presentation that sample preparation is the most customizable area of the Illumina process. Pretty much you can adapt the basic protocol to meet the goals of any project. As long as you end up with a library that has adapters on either side and the right conformation, you can almost get there any way you want. And again, sample tracking is hugely important for any size laboratory, small or large. In the high throughput world, if you can barcode your tubes and your plates and have an automated LIMS tracking system, that's the best. But even at low throughput, if you do Excel sheets, databases, just tracking things makes life a lot easier. And the Broad here, we developed many modifications to the standard protocol. We use Covaris for shearing, and we've optimized that for various size ranges. We've implemented the Ampere SPRI beads for automated cleanups and to perform gel-free size selection. If we do have to do a gel, we've optimized our gel conditions to be able to get really tight size deviations. And then we've also developed some high diversity protocols to get deep coverage libraries that have billions of molecules at the end. And of course, library quality monitoring is hugely important because you want to make sure that only the highest quality libraries make it to the sequencer. Even though library construction is very laborious, it actually is way cheaper in the grand scheme of things than sequencing. So you really don't want to put junky libraries on a sequencer. You want to sort of take the time to make sure that they're exactly what you want them to be before sequencing them. What you'll see in the lab section of this module is some in-lab sample tracking methods and tools that we use. You'll also see a demo of the Covaris E210. You'll see some gel cutting tips and tricks, as well as the special gel cutting tool we've developed. You'll see some library QC methods and some example data. You'll also see a demo of how SPRI works in the lab and an overview of some of the automation tools that we use here at the Broad. So thanks very much for your time, and I hope that you can take some of the stuff that we've talked about here and apply it to library construction in your own labs. Thank you.